In this video, we'll look at the Cerna paper treatment of the relationship between the regulation of stomata by the HT1 protein and the regulation of stomata by abscisic acid. If we look at the left-hand pair of bars, we'll first note that the y-axis is stomatal aperture. This is the same thing as pore size. So this describes the space between the guard cells through which water vapor can diffuse during transpiration. Now, the two bars represent wild-type plants. Control is in the absence of abscisic acid, and ABA-treated plants in the presence of abscisic acid, after abscisic acid treatment. And you'll notice that abscisic acid, as expected, closes stomata somewhat. Not very much abscisic acid was used, otherwise these stomatas would be tightly shut. But you notice something else too, that when compared to the HT1-2 starting stomatal opening, wild type are wide open. And this tells us something about the conditions in which this experiment was conducted. So this tells us that the experiment was conducted under low CO2 conditions. Otherwise, these bars would start out at the same level. Okay, abscisic acid, as we know, closes stomata. Now in this HT1-2 mutant, you lack. So remember, this has, yes, it's got HT1 function, and here, no HT1 function. That's the difference between these two plants. At low carbon dioxide conditions or high carbon dioxide conditions, any conditions, the plant is going to have relatively closed stomata to start with. But see here that even in these relatively closed situations, abscisic acid continues to function. So yes, abscisic acid reduces stomatal opening even when HT1 is not present. What does that tell you? That tells you that HT1 isn't required for abscisic acid closure of stomata. Now you may think at first, oh, but this is sort of comparing apples to oranges. Why didn't they use high carbon dioxide? Why not start with the control and the mutant bars in the same place, which would have occurred if they had used high carbon dioxide environment to carry out this experiment? But this is a much more clever setup. So the HT1 is not required for the closure of stomata by abscisic acid. That's pointed out by this comparison. But by doing it at low carbon dioxide environment, you can see that HT1 is active with or without abscisic acid being present. So HT1 isn't required for abscisic acid regulation. You see that. But it's also true that HT1 is still important even when abscisic acid is present, right? There's still a difference mediated by HT1 even in the presence. So abscisic acid isn't shutting down HT1. HT1 still functions when abscisic acid treatment occurs. That's why they did it in this high carbon dioxide environment.